evening viewer. Welcome to our Glory Globe TV viewers. A most amazing news for all of you. Yeah, our gut. Our gut is home to trillions of bacteria which play a crucial role in our health and disease. A recent study by researchers at EMBL Heidelberg reveals that this gut bacteria can trigger significant molecular changes in one of our most vital organs, the brain. Published in Nature Structural and Molecular Biology, the study is the first to demonstrate that gut bacteria can influence how proteins in the brain undergo glycosylation, a process in which carbohydrates modify protein. This breakthrough was made possible by a newly developed method called DQGIYCO, which enables researchers to analyze glycosylation with greater scale and precision than ever before. Proteins. Yeah, proteins are the workhorses of our cells and their main building blocks. Sugars or carbohydrates, on the other hand, are among the body's main sources of energy. However, the cell also uses sugars to chemically modify proteins, altering their functions. This is called glycosylation. Yeah, glycosylation can affect how cells attach to each other, adhesions, how they move, motility, and even how they talk to one another, communication. Explain. Clement Portal, first author of the study, and Savitsky team research and scientists. It is involved in the pathogenesis of several diseases including cancer and neuronal disorders. However, glycosylation has traditionally been notoriously difficult to study. Only a small portion of proteins in the cell are glycosylated, and concentrating enough of them in a sample for studying a process called enriching trained to be laborious, expensive and time consuming. So far, it is not been possible to do such studies on a systematic scale in a quantitative fashion and with high reproducibility and similarly called reproducible, said Mikhail Savitsky, team leader, senior scientist and head of the proteomics core facility at EMBL Heidelberg. These are the challenges we managed to overcome with the new method. DQ Xyloco uses easily available and low cost laboratory materials such as functionalized silica beds to selectively enrich glycosylated proteins from biological samples, which can then be precisely identified and measured. Applying the method to brain tissue samples from mice, the researchers could identify over 150,000 glycosylated forms of proteins, that is called proteoforms, an increase of over 25-fold compared to previous studies. The quantitative nature of the new method means that researchers can compute and measure differences between samples from different tissues, cell lines, species, etc. This also allows them to study the pattern of microheterogeneity, the phenomenon where the same part of a protein can be modified by many, sometimes hundreds of different sugar groups. Amazing. BN's exclusive report, Glory Globe TV, Delhi, Bharat.